How y'all doing? Good. Great in the back. Average. Tired. Tired. Massive weekend plans. You're going to get on top of all that studying, right? No. Yes. Okay. One yes. That's good to hear. Okay. I thought we would start with a song. That'll put us in a good mood to start today. And the song that I have for you is the one I was looking for at the end of yesterday. It's called When Acids Get Oxidized. So please join me. The fatty acids carried by CoA are oxidized inside the mitochondria to get to there, as you have seen, by hitching rides on carnitine. Then it goes away when acids get oxidized. Electrons move through membranes. Yes, it's true, it's true. They jump from complex 1 onto CoQ. The action can be quite intense when building proton gradients, and it's good for you when acids get oxidized. The protons pass through complex V, you see. They do this to make lots of ATP. The mechanism, you should know, goes through the stages LTO. So there's energy when acids get oxidized. Okay. Now you guys have all that down, now that we've got the song down, right? So I finished up last time talking about fatty acid oxidation and ketone body metabolism. And I hope that you appreciated the importance of ketone bodies and that overall sort of rescue scheme for cells, because for the body, because what it allows the body to do is provide energy when glucose isn't available. That's really why ketone body metabolism is important. The brain and eyes are very big users of ATP. They're very big users of carbohydrates. And so when carbohydrate levels fall, these ketone bodies become all the more important. I want to spend some time, in fact, we're in pretty good shape with where I want to be with respect to the lecture. So I want to spend some time talking about fatty acid biosynthesis and then metabolism of other lipids, and that's primarily what I'm going to be talking about today. Kind of like you saw with gluconeogenesis, the pathway for making fatty acids kind of overlaps with the breakdown of fatty acids. We'll see that there's differences, but the chemistry itself is very, very similar, and you're going to love the enzyme that does this. And I'll tell you about that in just a bit. Okay? So I start off by pointing out something important about fatty acid synthesis. Fatty acid synthesis occurs in the cytoplasm. Fatty acid oxidation was occurring in the mitochondrion. So we had to move the fatty acids into the mitochondrion. Well, this turns out to be very good because there's a spatial separation of synthesis and breakdown in the cell. So we don't have to worry so much about one process going on at the same time as the other process is going on because they're spatially separated. Okay? When does fatty acid synthesis occur? Okay? Well, fatty acid synthesis is going to occur when the cell has lots of energy and when the cell has lots of NADH. Okay? So we talked about respiratory control the other day, and I painted a picture for you about what happens when I'm out in my backyard, sitting in my Chase Lounge, drinking beer, eating pizza, and watching the world go by. Okay? Well, what's happening at that point? What's my level of ATP, did we decide? We said my level of ATP was high, right? What's my oxygen use? Low. I'm not burning anything up. I'm not doing anything, so my oxygen use is low. When my oxygen use is low, what's my NADH level? High. Okay? Now, we're going to see that NADPH turns out to be needed for synthesis, but NADH levels being high also means that NADPH levels will be high. Okay? Well, let's think about what this means. If NADH levels are high, what happens to the citric acid cycle? It stops, right? And if the citric acid cycle stops, where is it going to stop? it's going to stop before any reactions that require NAD. The very first of those in the cycle that we talked about was the isocitrate dehydrogenase reaction, where isocitrate is being converted into alpha-ketoglutarate. Okay? That's the first reaction in that cycle that needs NAD. So if there's no NAD, or very little NAD, the first thing that's going to accumulate is isocitrate. Correct? And isocitrate is the product of the reaction that converts citrate into isocitrate, right? That's the aconitase reaction. So if isocitrate concentrations are high, citrate concentrations are going to be high. And that turns out to be a very big signal, as we will see, for fatty acid synthesis. When citrate levels inside of the mitochondrion rise, citrate gets kicked out of the mitochondrion into the cytoplasm. There's actually a shuttle that moves citrate out of the matrix of the mitochondrion and into the cytoplasm. When citrate gets out into the cytoplasm, it gets broken down into acetyl-CoA and oxaloacetate. So what citrate has done is it's sort of carried on its back this acetyl group that becomes acetyl-CoA. Okay? Well, why is that important? Well, it turns out acetyl-CoA is necessary for the synthesis of fatty acids. So we've started a process of delivering acetyl-CoA out to the cytoplasm so we can make fatty acids. And when is this going to happen? This is going to happen when I'm sitting on my Chase Lounge, drinking beer, eating pizza, and not exercising. I'm going to get fat when that happens, okay? Because I'm going to make fatty acids. All right. Well, let's look at the process of making a fatty acid, starting with acetyl-CoA. Okay? So we're going to make a fatty acid starting with acetyl-CoA. You know that I told you that most fatty acids that exist biologically have even numbers of carbons. And so you may be inclined to think that acetyl-CoA is the building block for fatty acids, and it turns out the cell does an odd thing. 2 carbons get added each time, but not from acetyl-CoA. Got it? Okay. 2 carbons get added each time, but not directly from acetyl-CoA. There's an intermediate called malonyl-CoA, and then there's another intermediate after that I'll tell you about. The first intermediate is malonyl-CoA. And this reaction shows you how it is that malonyl-CoA is made. Acetyl-CoA is combined with bicarbonate, which is just a source of the carbon, a carbon dioxide as it were, using the energy of ATP to make malonyl-CoA. Well, this is a carboxylation, and we've learned that carboxylations use the coenzyme of biotin. The enzyme that catalyzes this is the most important enzyme for regulating the synthesis of fatty acids. Okay? This enzyme, I'm going to give you the name. You need to know this. It's acetyl-CoA carboxylase. Acetyl-CoA carboxylase. So it's taking a 2-carbon acetyl-CoA and making it a 3-carbon malonyl-CoA. Now, I said this enzyme is the most important enzyme for regulating fatty acid biosynthesis, and I'm going to tell you how it accomplishes that. Okay? First, what turns this enzyme on? Well, one of the things that turns this enzyme on is citrate. So as citrate is getting kicked out into the cytoplasm, some of it's going to bind to this enzyme and turn this enzyme on. What turns this enzyme off? What turns this enzyme off is palmitoyl-CoA. We'll see today that that's the last product of this process that we're starting. So the end product of this pathway inhibits the first enzyme, which means that this is a feedback inhibition. So palmitoyl-CoA, I'll spell it for you, Palmitoyl-CoA. Palmitoyl-CoA is an inhibitor of acetyl-CoA carboxylase. This enzyme turns out to be regulated in another way. And you begin to get the impression that when you have multiple ways of regulating an enzyme, like we saw with glycogen phosphorylase, when we see that there's multiple ways of regulating an enzyme, that enzyme must be pretty important to control, and it is. Okay? The other way in which this enzyme is regulated by phosphorylation and dephosphorylation. Okay? Phosphorylation and dephosphorylation. If we put a phosphate onto this enzyme, we turn it off. If we take the phosphate off of this enzyme, we turn it on. Okay? I saw a couple frowns when I said that we put a phosphate on, we turn the enzyme off. Putting a phosphate turns on some enzymes, turns off other enzymes. It turns off this enzyme. Okay? All right. There's the actual reaction. We're not going to worry about that. Okay? And here is what's happening in this pathway I'm going to describe to you. Okay? These are the steps in the synthesis of a fatty acid. You saw in the breakdown of a fatty acid that there were 4 steps in breakdown. The first step was a dehydrogenation that made a double bond, that trans double bond. The second step was addition of water, okay, across the double bond. The third step was the oxidation of the hydroxyl to make a ketone group on carbon number 3. And the fourth step was that thiolase cleavage that broke off that 2-carbon acetyl-CoA. It turns out that the pathway I'm getting ready to describe to you takes those 4 steps and reverses them. Instead of splitting off a 2-carbon piece, the first step is addition of a 2-carbon piece. But before I get to that, I need to tell you something that's different about fatty acid synthesis. Okay? Fatty acid oxidation occurred when the intermediates were linked to the CoA, coenzyme A. Fatty acid synthesis occurs when the intermediates are linked to a molecule called ACP. ACP is an acronym for acyl carrier protein. So that malonyl-CoA that we made has to have the CoA replaced by an ACP. Everybody with me? In the process of synthesizing a fatty acid, 2 of the carbons of malonyl-ACP will get added to a fatty acid chain that is even numbered. I'm going to repeat that. In the synthesis process, 2 carbons of malonyl-ACP will get added to a fatty acid that has an even number of carbons. So if we start with something that has an even number of carbons, and we add 2 carbons to it, and we keep doing that, we're going to end up with something that has an even number of carbons, even if we start with something that has 3 carbons. Well, that's what's actually going on in this process. Okay? Every time I look at this, I see another mistake, and there's a mistake right there. Okay, got to fix these. All right, here's the first step in the process of making a fatty acid. We've already made the malonyl-ACP. I said we add 2 carbons of a malonyl-ACP, okay, to a fatty acid that has an even number of carbons. That should actually be an ACP there. I'm not quite sure why that's not ACP. In any event, here is the 3-carbon malonyl-CoA, and the starting material that we have is a 2-carbon acetyl-ACP. A 2-carbon acetyl-ACP. 2 of these carbons are going to join with that. We're going to split out a carbon dioxide in the process. Okay? So here we go. Combine this with this. We're splitting out a carbon dioxide. And now we have a 4-carbon intermediate that you can see up here. So 2 carbons plus 2 carbons gives us a 4-carbon molecule. That 4-carbon molecule has a ketone on position 3, just like was in fatty acid oxidation. In fatty acid oxidation, we went from this 4-carbon molecule to split it in half to get 2 acetyl-CoAs. In this case, we're taking 2 molecules and putting them together to make a 4-carbon molecule. So we're reversing the process. It's happening now on ACP instead of happening on CoA. But essentially, we're reversing the process. We've got a ketone at carbon number 3. In order to have a fatty acid, we have to have all carbons and hydrogens. So we've got a carbon and an oxygen out here. We want to make sure we've got carbons and hydrogen out there. That means, therefore, that we need to reduce that ketone that's on carbon number 3. And that's the opposite of what happened in fatty acid oxidation, because in fatty acid oxidation, we went from a carboxyl to a ketone. Now we're going to go from a ketone back to a carboxyl. That's a reduction that's happening. And that reduction that happens as a result of catalysis by an enzyme whose name I'll talk about later. As I said, you're going to like the enzyme. Okay? But it also requires electrons from NADPH. NADPH is the source of electrons. Well, at this point, we've got a hydroxyl. Remember I told you when we looked at the stereochemistry of the hydroxyl in oxidation, I said note that it's an L, because in synthesis, you're going to see the hydroxyl is in a D configuration, and that's what that is here. In fatty acid oxidation, to get to that hydroxyl, we had to add water across a double bond. In fatty acid synthesis, we're going to pull water out and make a double bond. And that's the next step. Pulling water out and making a double bond, and yes, that double bond has a trans configuration, just like fatty acid oxidation did. We're almost there. The last step in this process is adding hydrogens and protons and electrons across that double bond. That is a hydrogenation. And by the way, don't confuse hydrogenation with hydration. Hydrogenation means you're adding hydrogens. Hydration means you're adding waters. Right? Okay. So we are adding hydrogens and protons and electrons across this. And look what we end up with. We end up with a molecule now that has 4 carbons. Everything beyond that carbonyl carbon has carbons and hydrogens in it. And this 4-carbon intermediate becomes the even-numbered fatty acid that gets added to on the next cycle. So we repeat this process. We'll get a fatty acid that has 6 carbons. We repeat that, we'll get 8. We repeat that, we'll get 10. We repeat that, we'll get 12. We repeat that, we'll get 14. I will stop in a minute. We repeat that, we'll get 16. And at that point, we stop. So this part of fatty acid synthesis only goes to 16 carbons. That is palmitoyl-CoA. That is palmitoyl-CoA. Now, I haven't given you any enzyme names. The only enzyme name I gave you was that one that made the malonyl-CoA. That was acetyl-CoA carboxylase, right? You'll see each of these enzymes have names going up here for all these reactions. And no, I'm not going to ask you to memorize those names, because all of those enzymatic activities are contained in the same multi-subunit protein that has the very complicated name of fatty acid synthase. So when I ask you what enzyme is involved in assembling the fatty acid, all you have to tell me is fatty acid synthase, S-Y-N-T-H-A-S-E. Okay? Okay, questions about that? No questions? Was I that clear, or are you that tired? And how many times have I asked that question this term? Okay. Okay. That process is occurring in the cytoplasm. This shows you something interesting. I told you that in fatty acid synthesis, we had an acyl carrier protein, ACP, in place of CoA. If we look at the ends of those, we see that they are virtually identical. The only difference is ACP is attached to a protein, and CoA is attached to AMP. Okay? That's the only difference between the two. Okay. And no, I didn't give you that name. I didn't say that name, so you're not responsible for that name, right? All right. You ready for another song? Okay. So I have a song that describes this in the same tune. So you should be really loud and boisterous in being able to sing this one. It's to the same tune, and it's called When Acids Are Synthesized. Okay? The 16-carbon fatty acid palmitate gets all the carbons that it needs from acetate, which citric acid helps release from mitochondria matrices. Oh, a shuttle's great when acids are synthesized. What did I do? Carboxylase takes substrate and it puts within dioxycarbon carried on a biotin. CoAs all gain a quick release replaced by larger ACPs, and it all begins when acids are synthesized. A malonate contributes to the growing chain. 2 carbons 7 times around again. For saturated acylates, there's lots of NADPH that you must obtain when acids are synthesized. Palmitic acid made this way all gets released. Desaturases act to make omega-3s. The finished products, big and small, form esters with the cholesterol. So you get obese when acids are synthesized. That's the bad news at the end, guys. Okay. Now we talk about metabolism of other lipids. I can tell you guys are just dying to jump into this. What do we do to liven this up? Come on, guys. Extra credit. Well, let me put it this way. I might be more inclined to give extra credit if I felt you were more engaged. How about that? Alert, asking questions, thinking, participating, not just going, Ahern, fill my head, right? So maybe, we'll see, okay? All right. I should have done the beer today is what I should have done. All right. Well, we've broken down fatty acids. We've made fatty acids, right? But there are other lipids besides fatty acids, and so we've got to think about those. Fats, and by the way, one of the most common things I see students confuse on exams, I keep saying this, but a very common thing students confuse on exams are fats and fatty acids. They're not the same thing. Fatty acids go into making a fat, but fats and fatty acids are not the same thing. Don't confuse them. Okay, well, let's talk about the synthesis of fats. Fats, of course, are triacylglycerols. And the synthesis of fats is actually very simple. Okay? Very simple. Where did phosphatidic acid come from? Where have you seen phosphatidic acid previously? What was used? What used phosphatidic acid to make something else? Anybody remember? There's a category of molecules that I've talked about in class that use phosphatidic acid as a precursor. There's a 2-point extra credit. What? Ketones? No. Not carbohydrates. What's that? Not nucleic acids. You guys are guessing here. Phospholipids. Bingo. Right? Glycerophospholipids, because think about this. Look at the structure of this. What is the backbone of phosphatidic acid? It is a glycerol. There's the lipid part. Those are the fatty acids. And there's the phospho part. Glycerophospholipids, also known as phospholipids, right? Okay. Phosphatid, and you get 2 points extra credit, so you'll have to see me afterwards about that, all right? There's our starting point. Now, it's going to be really hard for the next part. The next part is we're going to clip that phosphate off, and then we're going to put a fatty acid on its place, and we've made a fat. So phosphatidic acid turns out to be a branch point between the synthesis of phospholipids and fats. Okay? Turns out to be a branch point between the synthesis of phospholipids and a fat. Remember that a triacyl glycerol is the same thing as a fat. And the difference between a fat and an oil is only its melting temperature. Something that's an oil is a fat that's liquid at room temperature. Something that's a fat or whatever is something that's a solid at room temperature. Yes, ma'am? A triacyl glycerol is the same as a triglyceride. Yep. You may use triglyceride. Okay? All right, we've made a fat. What else have we made? Okay. Well, I said that phosphatidic acid was a branch point, so if we can make fats from phosphatidic acid, we can also make glycerophospholipids from fatty acids, and that's what we see here. Now, this seems a little bit more of a convoluted pathway than we saw in making a fat, and I don't want to overwhelm you with structures and figures here, and I'm not going to expect detail in terms of your memorizing things with this, but I will show you a couple of interesting things that happen with it. First of all, we start with phosphatidic acid because that's the branch point. And phosphatidic acid is combined with CTP to make CDP diacylglycerol. That's an important molecule because it's an intermediate in the synthesis of a lot of the glycerophospholipids. CDP diacylglycerol. I'd like you to look to see what happens in this process, and maybe you can tell me about why CDP is used. Okay? We see that CDP gets put onto this molecule right here, and then in the next reaction it comes off as CMP, meaning cytidine monophosphate. One of the phosphates stays behind, and we replace it, in this case, with a serine. What function did that CDP serve? You've seen a reaction like this already. It makes the reaction more favorable. How? What was the name I gave to that molecule? What's a molecule that has a lot of energy, that uses the energy to donate a part of itself to something else? What's that? Activated intermediate, right? Okay. This is an activated intermediate. Now with this, we see sort of closure of the use of all of the nucleotides for energy purposes. ATP was used in general for energy. GTP was used for protein synthesis. UTP was used for glycogen synthesis. And now you see CTP being used for glycerophospholipid synthesis. There's a very important reason why that happens. Okay? All of these provide energy, and balances of all of these balances are important. Okay? And cells that are going to divide need all of these in order to move forward. If the cell is low in energy, it's not going to be able to make the molecules that it needs, like carbohydrate, like a glycogen. It's not going to be able to make things for membranes like this. It's not going to be able to do protein synthesis, and it's not going to have ATP for everything else. By using nucleotides as their indicator, the cell has a very good idea about whether there's sufficient energy to go forwards. Okay? whether there's sufficient energy. When there's plenty of energy, all these things will get made, and the cell says, okay, now it's time to go. Okay, well, we've just made a phosphatidyl compound. In this case, we made phosphatidylserine. I can convert that into phosphatidylethynolamine. I can make phosphatidylcholine. I can make all kinds of phosphatidyl compounds. And I'm not showing you all this to get you to memorize all the different things that are made. But I do show you this so that you understand that CDP, diacylglycerol, is an important intermediate, and it's an activated intermediate for making glycerophospholipids. If you understand that, you understand everything that you really need to know in this figure. Okay. Here's some more of them being made. And you can see that CDP comes up in other ways. In the case you saw before, CDP was linked to diacylglycerol. Here it's linked to ethanolamine, and you accomplish the same goal. But in each case, you have to use the energy of CDP to make the glycerophospholipid. Okay? Very important point. All right. And even more. Okay? All right. Sphingolipids, we've talked about them already. And sphingolipids also are important compounds for making membranes. The synthesis of sphingolipids requires two things. One, it requires palmitic acid, the very thing that fatty acid synthesis just made. Okay? Second, it requires the amino acid serine. So you can see palmitoyl-CoA donating a palmitic acid. There's serine, and we make the first of the sphingolipids. Okay? So those two things are necessary for making sphingolipids. Oh, by the way, one thing I didn't mention, and I should mention about the fatty acid synthesis, and I apologize for that, is I told you how to get to palmitic acid. Palmitic acid had 16 carbons. But we have fatty acids in our cells that are longer than 16 carbons, and the cell has to be able to make those. And moreover, what I've shown you here is the synthesis of a saturated fatty acid. Okay? I haven't shown you the synthesis of unsaturated fatty acid, and I know you want to see it. That's a joke. You're really wanting that extra credit, aren't you? When I got to pull it out of you like that. Okay, no. So it's actually very simple from our perspective how this happens. All right. The synthesis of fatty acids longer than 16 does not occur in the cytoplasm. It occurs in the endoplasmic reticulum. Synthesis of fatty acids longer than 16 carbons occurs in the endoplasmic reticulum. The enzymes that catalyze those, you're going to love the names of them, they're called elongases. E-L-O-N-G-A-S-E-S. So elongases are found in the endoplasmic reticulum. How do we get unsaturated fatty acids? Well, to make unsaturated fatty acids, we have to make double bonds. And so we have a set of enzymes known as desaturases that do that. Okay? We have enzymes known as desaturases that do that. And the desaturases we've already talked about can't make double bonds in animals further away than carbon 9. So if we need things like linoleic or linolenic, which have double bonds out past carbon number 9, they must be in our diet, which is why we must have them as essential fatty acids. Desaturases are also found in the endoplasmic reticulum. Okay. I'm going to do cholesterol biosynthesis, and then we'll do something else. All right. Here's the structure of cholesterol, and it shows you M's and C's. What are these M's and C's? Well, it may surprise you, but cholesterol in the body is made completely from acetyl-CoA. Completely. Acetyl-CoA is a 2-carbon molecule and it goes together to make this monstrosity up here. The M on here comes from the methyl group of the acetyl-CoA, and the C comes from the carbonyl of the acetyl-CoA. It's pretty remarkable. We're going to see how that happens, and that's even more remarkable in that it may seem complicated, but you're going to see there's some great simplicity to it. So what we're talking about now is how the body makes cholesterol. Okay? How does that happen? Well, the body starts with, there's the M and there's the C, by the way. Okay, so you can see what I'm talking about. The body starts with 3 molecules of acetyl-CoA. And it makes a molecule called mevalonate, which has 6 carbons. Where have you seen mevalonate before? Anybody remember? Where did you see HMG-CoA before? I said there was a pathway that overlapped with cholesterol biosynthesis. That pathway is ketone body metabolism. That was Wednesday. That was 48 hours ago. Okay? Ketone body metabolism and cholesterol metabolism overlap with HMG-CoA. In ketone bodies, they go a different direction, but HMG-CoA goes to mevalonate. Okay? And this is catalyzed by a very important enzyme whose name you're going to need to know. The enzyme is called HMG-CoA reductase. HMG-CoA reductase catalyzes the formation here of mevalonate. It is an enzyme that is feedback inhibited by the end product of its pathway, meaning HMG-CoA is inhibited by cholesterol. As cholesterol levels build up, HMG-CoA becomes inhibited if everything's working right, which means the body will slow down synthesis of cholesterol when cholesterol levels get high. Well, how does a person have high cholesterol levels then? Well, there's a variety of ways a person can have high cholesterol, and I'm actually going to talk about that in the lecture on Monday. I'm not going to talk about that today. Okay? All right. HMG-CoA is the most important regulatory enzyme for this pathway, and as I'll show you in a minute, that enzyme is a target for drugs. So if you know somebody taking cholesterol-lowering drugs, they most likely are taking compounds that inhibit HMG-CoA reductase. There's a class of compounds called statins, S-T-A-T-I-N-S, that inhibit that enzyme, and we'll talk about those in a bit. Okay? All right. Mevalonate got 6 carbons. You break off one of those carbons, you get 5 carbons. That creates a molecule who has a general name called isoprene. Isoprene simply means 5-carbon intermediate. Okay? There are 2 different 5-carbon intermediates, and I'm going to have mercy on you and not give you the names to know. They're both isoprenes. Okay? Those 2 isoprenes can combine together. If you take a 5-carbon molecule and you combine it with a 5-carbon molecule, you make a 10-carbon molecule. This is very good. If you take a 10-carbon molecule and you add another 5-carbon molecule to it, you make a 15-carbon molecule. You guys start to get into this. And if you take 2 15-carbon molecules and you join them together, you make a 30-carbon molecule, and that is squalene. You've just started with a 2-carbon molecule, and you've gotten to a 30-carbon molecule. Squalene can be rearranged in many steps by twisting bonds and so forth to get to cholesterol. Okay? I will show you something here. Okay? Here's acetyl-CoA, beta-hydroxy, this HMG-CoA here. There's mevalonate. That's what I've already talked about. Okay? here are the reactions that get you to where I just described to you. There's the molecule. There's one of the 5 carbon molecules. There's the other, and I haven't said the names yet. I put 2 together. I made a 10. I took the 10, added a 5, and I made a 15. I took the 15s together and made a 30. You should know the names of the 10s and the 15s. The 10 is called geranyl pyrophosphate, G-E-R-A-N-Y-L, geranyl pyrophosphate. The 15 is called farnesyl pyrophosphate. It's on the screen there. What's that? Yeah, I'll spell it for you. F-A-R-N-E-S-Y-L. I can't zoom, unfortunately. F-A-R-N-E-S-Y-L. At least not very much, I don't think. Oh, there it does go a little bit. My own magical slide. I didn't even know that. So, okay. All right. We've made squalene. I told you there was some twisting of bonds to make something like cholesterol. And this is really interesting to me. You guys probably don't give a hoot about it, but I care about this, right? I can take that 30-carbon squalene, and I can rearrange those bonds into something very easily that looks like cholesterol. And it looks like cholesterol, and you would think it's a very simple pathway to go from lanosterol, which is this compound here. It's the first, what we call cyclic intermediate, down to cholesterol. But to get from lanosterol to cholesterol takes 19 steps. When I was a graduate student, I had to memorize every one of those 19 steps. And I vowed that I would always make my students learn every one of those 19 steps. Now you're wondering how serious he is, right? I've never made anybody memorize them because I like living by the end of the class. But I had to do that at one point. That was a real pain, okay? 19 steps to cholesterol, all right? That's a lot of steps. The last thing I'm going to show you today are the statins, which are the compounds I mentioned before, okay? The statins resemble HMG-CoA. They also resemble mevalonate. And they bind to the enzyme and competitively inhibit HMG-CoA reductase. They competitively inhibit HMG-CoA reductase. Now, I've got one last song, and depending on how loud people sing, we'll see about extra credit. And fortunately, it's to the same tune we've done the last 2. So I know you know the tune. Okay? It's called... Oh, what happened to my song? There it is, to make a cholesterol. Ready? Some things that you can build with acetyl-CoAs are joined together partly thanks to thiolase. They come together, 1, 2, 3, 6 carbons known as HMG, and you're on your way to make a cholesterol. To synthesize a mevalonate in the cell requires reducing HMG-CoA as well. The enzyme is a reductase controlled in allosteric ways when the cell's impelled to make a cholesterol. The mevalonate made in metabolic schemes gets decarboxylated down to isoprenes. They're linked together willy-nilly to build a PP geranyl in the cell's routines to make a cholesterol. A single step links farnesyls, but that's not all. The squalene rearranges to lanosterol. From that, there's 19 steps to go before the sterols apropos, which you must recall to make a cholesterol. The regulation of the scheme's complex in ways inhibited by feedback of the reductase. And statins mimic, so they say, the look of HMG-CoA. So we sing their praise and not make cholesterol. You guys really want extra credit. I didn't get any cooperation until the very end, but all right, it's Friday. What should the extra credit be? I like the portraits, by the way. That was very good. I know. I think you should draw the portrait of the TA who comes to the class every day, who's now leaving.